Welcome. We are going to let about a minute go so that we can allow others to log in to this webinar, but you are in the ICER and California Technology Assessment Forum webinar today on lecanemab for early Alzheimer's disease. We'll be starting in about 1 minute. Thank you.
Welcome, everyone. Good morning. Thank you for attending today's public meeting of the California Technology Assessment Forum, CTAF, a partner of ICER, the Institute for Clinical and Economic Review. I am Rena Fox, chair of CTAF and of today's forum. Today, we will be discussing lecanemab for early Alzheimer's disease, reviewing the evidence, hearing testimony from stakeholders, and deliberating over the comparative clinical effectiveness, other potential benefits, and long-term value for money. At the beginning of each meeting, we ask each of our CTAF members to disclose any financial relationships with industry. Every member of the CTAF panel who is present has met the ICER conflict of interest policy. Would each CTAF member introduce themselves, their work, and note whether they have any updates to this declaration? I will call on each CTAF member, and I will begin. I am Rena Fox, professor of medicine. I'm a physician and clinical researcher in the Division of General Internal Medicine at UCSF. I have no updates. Felicia Cohn.
I am Felicia Cohn, Director of Bioethics for Kaiser Permanente in Orange County, and I have no updates.
Thank you. Robert Collier.
Hi, I'm Bob Collier. I have about 30 years as a patient advocate in the area of medical research. I have no updates on conflict of interest.
Thank you, Bob. Sanket Dhruva.
Sanket Dhruva, staff cardiologist at the San Francisco Veterans Affairs Healthcare System and assistant professor of medicine at UCSF and a health services researcher. I have no updates to my conflicts.
Thank you. Kim Gregory.
Good morning, I'm Kim Gregory, Vice Chair and Professor, Department of OBGYN at Cedars-Sinai Medical Center, and I have no updated conflicts.
Thank you. Paul Heidenreich.
Paul Heidenreich, cardiologist at Stanford University School of Medicine, and I have no updates.
Jeffrey Hotch.
Good morning, I'm Jeffrey Hotch. I'm a professor of health economics in the Department of Public Health Sciences at the University of California at Davis. I have no updates.
Thank you. Jeffrey Klingman.
Morning, I'm Jeff Klingman. I'm chair of neurology at Kaiser Permanente in Northern California. No updates.
Thank you. Annette Langer-Gould.
My name is Annette Langer-Gould. I'm the regional lead for translational neuroscience at Southern California, Kaiser Permanente, and a practicing MS specialist and epidemiological researcher. And I have no updates.
Sei Lee.
Hi, my name is Sei Lee, professor of medicine in the Division of Geriatrics at UCSF. No updates.
Joy Melnikow.
Hi, I'm Joy Melnikow. I'm a physician and researcher in the Department of Family and Community Medicine at UC Davis. I'm a professor emeritus there, and I have no updates.
Elizabeth Murphy.
Hi, I'm professor of medicine at UCSF and chief of the Division of Endocrinology at San Francisco General Hospital. I have no updates.
Catherine Phillips.
Good morning, everyone. Catherine Phillips, professor of health economics, University of California, San Francisco. No updates.
Rita Redberg.
Hi, I'm Rita Redberg. I'm a cardiologist and professor of medicine and faculty in the Institute for Health Policy Studies at UCSF, and I have no updates.
And Tony Sowery.
Yes, hi, good morning. I'm Tony Sowery. I represent the National Patients Advocate Foundation. I have no updates or conflicts of interest.
I don't think I've forgotten anyone. Hopefully not. Thank you, CTAF. At this point, we will also ask our individual patients and clinical experts to introduce themselves and declare if they have any potential conflicts of interest. Doreen Monks.
Good morning. My name is Doreen Monks. I'm a retired neuroscience nurse practitioner and program director for the Stroke Center at St. Barnabas Medical Center in Livingston, New Jersey. At age 61, I was diagnosed with early onset Alzheimer's disease, and I have no conflicts of interest.
Thank you, Doreen. Russ Paulson.
Hi, I'm Russ Paulson. I am Chief Operating Officer at Us Against Alzheimer's. I have no personal disclosures. Institutionally, we are a nonprofit and we survive on donations. We get donations from a lot of people, including ASI and its competitors. We are also governed by a board of directors that has no reps from pharmaceutical companies.
Thank you. Dr. Victor Henderson.
Hi, Victor Henderson. I'm a neurologist who cares for people with dementia. I'm a professor of epidemiology and neurology at Stanford University, where I also direct an Alzheimer's disease research center funded by the National Institute on Aging. I have no conflicts.
Thank you. And Dr. Jason Karlowicz.
I'm Jason Karlowicz. I'm a geriatrician. I'm a professor at the University of Pennsylvania. I co-direct the Penn Memory Center where I care for patients, and I'm the associate director of our NIA Alzheimer's Disease Research Center. I have no additions to my disclosures that I gave.
Thank you, everyone. I would also add a reminder to everyone, whether it is later during the policy roundtable or during any public comment, that any individual making public comment announce any relationship with industry or other potential influences on judgment before you speak. We will also present this information publicly on slides. And at this point, I'm very happy to introduce Dr. Steve Pearson, President and founder of ICER. Steve.
Thank you, Rena, and good morning, everybody. And thank you to our participating patient experts and clinical experts. So let me do a brief introduction to today's meeting. You've already met, sorry, I should have advanced these slides before. You've already met our clinical experts and patient experts who will be with us throughout the morning when the evidence is discussed, when public comment is welcomed, and then very much at the center, at the heart of our policy roundtable later this afternoon. Why are we here today? We always want to start with the patient's perspective. And here's a quote: "I get frustrated when people question my diagnosis. Oh, you're fine and are just looking for attention. I get frustrated when I can't understand a joke and everyone else is laughing. I get frustrated when I don't remember somebody, but they remember me. It's mortifying. It's hard when I'm not sharp like I used to be." Now, this is one person living with early Alzheimer's disease. Many of us have personal or family experiences with this condition. We know how common it is. We know how devastating it can be. We know how diverse its effects can be on individuals and families. At the meeting that we had on aducanumab, I shared, my own family's history in which my mother died of Alzheimer's disease, and my father also had dementia. And both personally and professionally, again, many of us will understand how important it is and how the unmet need has been driving tremendous efforts to try to accomplish both prevention and treatment options to help many, many people around the world. So we're here to talk about that. And the day that a treatments receive FDA approval, there is again, very commonly a justifiable sense of celebration. There's a sense that, patients and families and clinicians have a new option, a new hope, if you will. And that's an important time to recognize the years of investment, of risk, of participation, again, by patients and families in the clinical trials, the many failures along the way that often accompany the arrival of a new treatment option. But other things happen that first day. When a drug is first approved, there are questions. There are questions about, okay, it's made it through the FDA, but what do we really know about the risks and benefits for us, for our family members, for broader groups of patients? because the evidence is always relatively new and immature, if you will, and there's a lot yet to be known. There are a lot of questions often too about how treatments fit into an evolving landscape of other options, whether they're current options or things that are just about to arrive in a few months or years. And there are important questions about, what are the reasonable pricing? Because the day that a drug is approved by the FDA, the company has the power to set a price. And the United States does not currently federally manage launch prices of new drugs. So this is a big question often. What happens, what are the reasonable costs to individual patients through their insurance systems? What are the reasonable costs and prices for the health system overall and for the government, which often picks up a large part of the tab, especially for treatments for patients over age 65. And we're also here today because we want to talk about the lessons that have been learned to the point where we are today, and what lessons might we be learning about what we need to do in the future. All of this is driving the reason that we're having the meeting today. And another element that has to be, I think, an important part of the backdrop for why we're having this meeting is because we can talk about, the costs and the benefits, the risks of treatments for Alzheimer's in its own context, but it has ramifications more broadly. Every new treatment, every new program that we introduce in the healthcare system has ramifications for other people, in part because they can help drive up the overall cost of healthcare. Now, this was brought home in a report last year from the Kaiser Family Foundation, in which they did a survey that demonstrated, and I think still shockingly, that approximately 100 million Americans are carrying medical debt today. And that includes real people, real families. That we can talk about the huge statistics, but just again, to put certain things in context, here are a few of the stories from that survey that were brought out in the public media. Marcus and Allison Ward on the left, and their children fell into medical debt when the kids were prematurely born, and they had expenses that were not covered by insurance, and others that were, but they had deductibles. And they ended up needing to sell their home and move to live with family, and are still in medical debt as a result of those costs. Sam and Ariane Buck from Arizona in the middle, you can say ironically perhaps, but his job was to get on the phone with Medicare recipients and to try to encourage them to sign up for Medicare Advantage plans. He was helping them, quote unquote, get health insurance when he fell into medical debt because he was underinsured and ended up in the ER and ended up with $30,000 of expenses that he could not cover. So he and his wife had to declare bankruptcy. Sherry Foy on the far right, she had very complicated abdominal surgery issues and was on an employer's kind of retirement plan that had a cap, and she too fell into medical debt and had to declare bankruptcy. Now, these are just individuals struggling with the system, and no one is suggesting that the cost of a single drug or even of all drugs are the primary driver of medical debt in this country, but they're a part of it. And every time we have a new treatment that costs anything extra in our system, we just have to be sensitive to the idea, the reality that real people struggle today to make ends meet with their medical insurance costs, with their premium costs just to maintain insurance. And our system is really requires us to take a step back and think about the broader balance here. And that balance perhaps is brought into even more sharp focus when we realize that the people who have medical debt in this survey, 1/7 of them had been denied care from a hospital or a doctor's office because of their medical debt. 2/3 reported cutting back on their own prescriptions or their own medical care as a result. And Black adults are 50% more likely than white Americans to be in medical debt today. So there are a lot of intersecting problems that we just have to keep in the back of our minds. And so as we keep our focus today predominantly on patients and families with Alzheimer's, wanting to get it right for them, we just have to remember that around them, around all of us, are families and others with whom we have to make sure that we create an overall innovative healthcare system that is affordable and equitable so that everybody can benefit, not just from today's innovation, but from the innovations of tomorrow. So that's why we're here today. And I'm glad you've had the chance to be introduced to the members of the California Technology Assessment Forum. Just a few more words about ICER, which is the convening host for today. Our funding, we keep on an updated pie chart on our website. You can see that 65% right now this year is coming from nonprofit foundations. The largest funders within that group are the Laura and John Arnold Foundation, the California Healthcare Foundation, the Commonwealth Foundation, and there are others. We do take approximately 25% of our overall revenue directly from life science companies and health plans and provider groups, but we use that money and kind of keep it separate for our ICER Policy Summit program, which brings them together to try to work collaboratively to look at how better we can generate evidence and apply it throughout the healthcare system. And we have about 10% of the rest of our funding that comes from subscriptions to our ICER Analytics cloud platform and from individual philanthropists. The ICER report today, that's the backbone in some ways of the discussion, starts approximately 8 months ago with a scoping phase in which we reach out to patients and patient groups, clinical experts, and manufacturers involved and other stakeholders to try to make sure that we get oriented to the diversity of the clinical experience, what patients think is important about value and the barriers to access that they're experiencing, the arc of the innovation, and other aspects that will really help us target and orient what we're doing. Then we do an internal staff job of looking at the evidence, and we're not just tied to looking at the published evidence. We work with the manufacturer to try to get the best up-to-date evidence we can. We work with patient groups to try to see if we can supplement the published, if you will, clinical trial data with patient-reported outcomes or any other helpful information. And we do a cost-effectiveness model built on that sum of information. Our draft report goes through several iterations of public comment and revision. And for this report in particular, we did have initial feedback from Us Against Alzheimer's, among others. Then we have an expert reviewer set. And among the clinical experts, we were fortunate to have specific comments from Victor Henderson and Jason Karlowicz, who are with us today. And all of this, report material is structured in a way, in a sense, to help support today's discussion, which in some ways is really talking about value. And in particular, long-term value for the money that would be spent. And to look at that, we do have to think about kind of a complex set of concepts that can build up to value or contribute to value. Starting at the bottom, we want to consider whether new treatments help us live longer. We also want to consider, some do, but some don't, and some treatments can help us have better health benefits by returning function to us or by having fewer side effects, slowing the decline, if you will, in certain functions, et cetera. And when we look at costs, we are not just looking at drug costs. We are looking at costs across the health system. So that if a treatment helps prevent future hospital, visits or doctor visits or procedures, we count that. And we also, in one version of our cost effectiveness, also look at costs outside the healthcare system to the extent that we can. The two buckets in blue, though, if you will, sometimes fall outside of what we can gather directly from clinical trial information. And so we try to really gain perspectives and data where possible on the benefits of a treatment that might spill over beyond health. And we'll talk more about the potential for treatments to help family members and individuals with their work or other aspects of their lives. And then there are sometimes, in particular, special social or ethical priorities, sometimes related to equity, health equity, sometimes related to the burden of illness, the severity of the condition that we want to try to factor into our thinking about value. So all of these components are part of the ICER report, and all of them will come into play today as we talk through this and try to create a transparent, more transparent process in which our society, if you will, and all the stakeholders involved can come together to talk about this, to seek that overall balance I talked about. So our agenda is fairly straightforward. We'll start with a presentation of the clinical evidence, then the economic model with time for interaction with CTAF, their questions, participation with the clinical and patient experts. Then we will have a formal public comment session that will start around 10:40 a.m. Pacific. We'll have lunch before noon on the Pacific time, and then come back for final deliberation and voting by the CTAF. And then we'll have one more break before we go to our policy roundtable, where we have some really important issues to talk about, because that's where we'll, if you will, kind of pivot away from talking further about the evidence to talking about what's going to happen in the healthcare system. How will this be covered? Will it be covered? Who's going to, pay for what? What, what kind of mechanisms of coverage are going to be considered by Medicare and others? How will clinical experts and patients share information so that we can get the right form of shared decision-making? All of those kinds of important health system, insurance, pricing, coverage, and future research questions will be covered then. We'll close with some reflections from the CTAF members themselves and adjourn by 3 p.m. Pacific time. So with that, thank you again for joining us, and let me hand it over to Dr. Grace Lin to begin the presentation of the clinical evidence.
Thank you, Steve. Good morning, everyone. I'm Grace Lin. I'm the Medical Director for Health Technology Assessment at ICER, and I'm also a Professor of Medicine and Health Policy at the University of California, San Francisco. And I'll be going through the clinical evidence for lecanemab today. Next slide, please. This presentation report was put together with the collaboration of my wonderful research team, including Abigail Wright, who's the Senior Research Lead at ICER, Serena Heron-Smith, who's Associate Research Manager at ICER, and Felucia Adbula, who's the Vice President of Research at ICER. We have no conflicts of interest relevant to this report. Next slide, please. I don't think I need to tell anyone here that Alzheimer's has a huge impact. It's a common disease with high human and medical costs. There are an estimated 6.5 million cases of Alzheimer's in the United States. And with this, there's a large caregiving burden, with 11 million unpaid caregivers providing 16 billion hours of care to those 6.5 million patients. It's also a very expensive disease, as you can imagine. The estimated direct costs for medical costs exceed $300 billion annually, and caregiving costs add another almost $300 billion annually to the cost of the disease. Next slide, please. Here we have the 5 stages and symptoms of Alzheimer's disease. There are 5 stages, each of which is defined by symptoms and functional impairment. The population we'll focus on is the mild cognitive impairment due to Alzheimer's disease population and the mild Alzheimer's disease population. While memory loss and impaired judgments are the hallmarks of MCI, when the symptoms start to interfere with everyday activities, such as having language problems, mood swings, or personality changes, patients get diagnosed with mild Alzheimer's. Next slide, please. We had the privilege of speaking with people living with Alzheimer's as well as caregivers to give us some firsthand insight into living with this disease. Some of the challenges that we heard were mentioned include coping with the diagnosis and changes in their lives, such as the loss of independence. One person told us that Alzheimer's made their world smaller because of the limitations on the things they were able to do. Caregivers mentioned that there's a lack of cohesive care after diagnosis, leaving persons with Alzheimer's and their caregivers to navigate the disease on their own. This leads to a tremendous amount of stress on the caregivers, many of whom are unpaid, and as a result, caregivers can suffer both mental and emotional tolls. Depression, anxiety, and disrupted sleep, for example, are some of the more common effects. Patient groups highlighted that there are health inequities in Alzheimer's disease, with a higher prevalence of Alzheimer's in Black Americans, differences in caregiving burden, and also a lack of culturally competent care. Finally, people with Alzheimer's were concerned that outcome measures used in clinical trials don't fully capture what's important to them, including a sense of independence and a sense of self, and maintaining their current quality of life. Next slide, please. The treatments for Alzheimer's disease focus on symptoms and slowing down decline, because as you know, there's no cure for this disease as of yet. Current treatment is primarily supportive care, which includes non-pharmacologic treatment, but also symptomatic drugs such as cholinesterase inhibitors and NMDA receptor antagonists. And then finally, there is a final category of disease-modifying agents. This class currently consists of monoclonal antibody infusions that aim to remove amyloid from the brain. And today we're going to focus on the newest anti-amyloid agent, lecanemab, and review both its clinical and cost effectiveness. Next slide, please. So let's dive into the clinical evidence. So the data we're reviewing today comes from Clarity AD, which is a large randomized placebo-controlled phase 3 trial. In this trial, there were approximately 1,800 people diagnosed with MCI or mild AD. The mean age of the population was around 71 years old, and more than 2/3 of the population had a mutation in the apolipoprotein E, E4 gene, or I'll call it ApoE for short, which is a gene that increases the risk of developing Alzheimer's. More than 3/4 of the participants identified as white, and their baseline cognitive score was on the CDR-SB, which I'll talk about in a moment, it was 3.2. Participants were randomized to either a 10 milligram per kilogram IV infusion of lecanemab or a placebo infusion every 2 weeks. This was on top of supportive care, which could include being on a symptomatic Alzheimer's medication, such as an acetylcholinesterase inhibitor. And about 50% of the participants were on a symptomatic medication at baseline. Participants in the trial were treated and followed for 18 months, and those are the results I'm going to discuss today. There is also an ongoing open label extension for the trial, and that will follow patients for up to 69 months. Next slide, please. Here we see the key outcomes. The key primary outcome is the clinical dementia rating sum of boxes, or CDR-SB. This is a scale that measures 6 domains of cognition and function, including memory, orientation, and judgment and problem solving, involvement in community affairs, home and hobbies, and personal care. Each domain is scored from 0 to 3, so you have a cumulative score of 0 to 18, and higher scores indicate more severe disease. There are also a number of secondary outcomes that were measured, both cognitive and functional scales, such as the ADAS-Cog14 and others, health-related quality of life scales, such as the EQ5D5L for patients, and also caregiver burden scores like the Zaret Burden Interview. And then there were also biomarkers measured, including amyloid, p-tau, and t-tau, which some of which were measured by positron emission tomography or PET scan, and some in the cerebrospinal fluid or the CSF. Next slide, please. So first, let's look at the effect of lecanemab on removing amyloid from the brain. As you can see in this graph, which is data from a subgroup of participants who underwent serial PET scans to look at amyloid in the brain, the lecanemab group, which is the yellow line, had a substantial decrease in the amount of amyloid in their brain. In fact, at 18 months, about 1/3 of the lecanemab group had enough amyloid removal to be labeled amyloid negative, which is they defined as less than 30 centiloids on the PET scan, compared with about 8% of the placebo group. And so I think we can see that lecanemab is very effective at removing amyloid from the brain. Next slide, please. In terms of clinical effect, for CDR-SB, we can see here in this figure, the horizontal axis represents time in months, and the vertical axis represents the mean change from baseline of CDR-SB, where larger changes represent greater worsening. You can see here that the yellow line, which is lecanemab, and the blue line start to diverge around 3 months of treatment. And after 18 months of treatment, there was a mean difference in CDR-SB score of 0.45 points, which represents a 27% slowing of decline between the groups. This difference was statistically significant. Next slide, please. As I mentioned, quality of life was also measured in the study using multiple quality of life scales. So you can see the first two scales at the top, EQ, the EQ5D, 5L, and the quality of life AD scales were participant quality of life scales. And then the two at the bottom, the quality of life AD and the Zaret Burden interview were caregiver quality of life scales. Changes in all of these scales were statistically significant, and like the CDR-SB, they favored less decline in the lecanemab treated group. As you can see, anywhere from 23 to 56% less decline over the placebo group, meaning that the quality of life did not deteriorate as quickly in the lecanemab group for both the participants and the caregivers. Next slide, please. There were a lot of other outcomes measured in the trial. Here are a few key secondary cognitive outcomes. In addition to the CDR-SB, there are scales such as the ADAS-Cog14, which is the Alzheimer's Disease Assessment Scale 14-item cognitive subscale. There was also the Alzheimer's Disease Composite Score and the Alzheimer's Disease Cooperative Scale measuring activities of daily living. Each scale measures a slightly different aspect of function, but if you look across the three panels here, the lecanemab group consistently performs better than the placebo group after 18 months, and all the differences were statistically significant. But the magnitude of benefit was relatively small in each case. For example, for the ADAS-Cog14 scale, there was a difference of 1.4 points on a 90-point scale. Next slide, please. Multiple biomarkers were also measured, including in the cerebral spinal fluid, plasma, and MRI. For the markers in the CSF and plasma, including t-tau and p-tau, we saw statistically significant reductions in levels in the lecanemab group compared with the placebo group, which would represent less of those markers, which represents an improvement. On the other hand, the MRI outcomes were mixed. There was less atrophy or less shrinkage in an area of the brain called the hippocampus, which is that shrinkage is commonly seen in Alzheimer's disease patients. On the other hand, there was a greater decrease in whole brain volume and a greater increase in ventricular volume in the lecanemab treated patients. And the decrease in whole brain volume and the increase in ventricular volume has been seen with other amyloid therapies and have prompted some concerns about the long-term effects of anti-amyloid treatments, as these changes are typically associated with worse progression of disease. Next slide, please. As you may know, anti-amyloid drugs have the potential to cause harm. In particular, the main serious side effect to worry about is ARIA, or amyloid-related imaging abnormalities, which can be in the form of brain edema or swelling, what we call ARIA-E for short, or brain hemorrhage or bleeding, also called ARIA-H. For both ARIA-E and ARIA-H, there were higher rates in the lecanemab group compared with placebo. Most of the ARIA was asymptomatic or mild to moderate in severity and occurred early in the treatment course, and most cases resolved within 4 months. However, almost 3% of patients experienced symptomatic ARIA-E, which includes nausea, headaches, confusion, and in severe cases can cause seizures and death. ARIA in general and severe ARIA were more common in patients with the ApoE mutation, particularly patients who were homozygous for the ApoE mutation. Also, while the deaths were no different between the lecanemab group and the placebo group in the core clinical trial, that's the 18-month trial, in the ongoing open label extension portion of the trial, there have been 3 reported deaths, and these deaths have been related to brain hemorrhage in patients given or taking anticoagulants. And in one case, the death is being investigated to be related to potentially severe ARIA. Next slide, please. So this review of the data for lecanemab has brought up some concerns, and I'm going to highlight some of these controversies and uncertainties in the data. I think it's clear from the data that lecanemab is very effective at removing amyloid from the brain, at least the amyloid that can be seen on PET scan. If the amyloid hypothesis is correct, then the clearance of amyloid should be closely correlated with an effect on cognition. But there have been some systematic literature reviews of anti-amyloid drugs that have shown inconsistency between amyloid removal and the slowing of cognitive decline. For example, in the aducanumab trials, one phase 3 trial showed an effective treatment on cognition and the other one did not. We don't have data from individual participants that to be able to correlate amyloid clearance with cognitive outcomes that would help us better answer this question. And even something like the proportion of patients who cleared amyloid and their outcomes would be helpful, since almost a third of the lecanemab group were amyloid negative at the end of the trial. Clinical experts also differ on whether the degree of slowing of cognitive decline, a difference of less than half of a point on the CDR-SB scale that was seen in this Clarity AD trial, reaches clinical significance. In the published literature, the minimum clinically important difference, or MCID, for CDR-SB is thought to be around 1 to 2 points, depending on whether you have MCI or mild Alzheimer's disease. Additionally, there are some notable differences in effect based on subgroups, with lecanemab having seemingly less effect in younger patients, women, and ApoE homozygous participants. We've also got concerns about the generalizability of the data to the population of US persons with Alzheimer's disease and to safety with real-world use. The clinical trial population was younger, less diverse, and had fewer comorbidities than the general Alzheimer's disease population. And so we don't know what the efficacy of lecanemab is in patients who don't fit these clinical trial eligibility criteria. We're also concerned that real-world monitoring of ARIA may be less stringent than in the clinical trial, which may heighten the potential risks of the drugs, particularly in patients who don't fit the clinical trial criteria and also who are on anticoagulants. Next slide, please. If lecanemab does provide a clinically significant benefit, the delay in progression could greatly impact the ability to achieve major health goals for both patients and caregivers, although that effect may be dampened by the complexity of the logistics of treatments, which is biweekly IV infusions and MRI scans for monitoring. Effective treatment could help heighten awareness and lead to more screening and diagnosis, and that could potentially decrease health inequities. For example, we know that African-American Hispanic patients tend to be underdiagnosed and diagnosed later, and therefore greater screening and earlier diagnosis due to having effective treatment could help decrease those disparities. Next slide, please. As always, we appreciate the public comments that we received on our report. We received comments on our use of MCID. Again, this is the minimum clinically important difference. We drew our MCID interpretation from published studies in the literature. Some experts debated whether the values we cited are appropriate and can be applied to the mean change in outcome, which is the what we have results from the clinical trial. This is because the concept of MCID was meant to be applied to changes within the individual patient. While there is heterogeneity in treatment response across patients, unless the distribution of outcomes in the trial was skewed, based on the difference we see, the mean difference we see in the CDR-SB, more than half of the patients in the trial would not have achieved MCID on an individual level. As I mentioned previously, it would be ideal to have individual data to confirm the proportion of patients who had differences in cognition that reached MCID. We also received a comment that the risk of ARIA is overstated. There was a protocol for successfully managing ARIA during the clinical trial, but it's not clear that patients receiving the drug in the real world settings will get the same level of clinical monitoring and MRI imaging that was done in the trial. We had some very helpful comments about the disproportionate impact on the African-American community with later diagnosis, more severe symptoms, and documented greater caregiver burden. And underrepresentation in clinical trials, and we've tried to reflect that in our revised evidence report. In summary, lecanemab shows statistically significant slowing of cognitive decline over 18 months of treatment. Whether this change is clinically significant is still a question that we are debating. ARIA remains a concern, particularly given the 3 reported deaths in the open label extension. And the clinical trial results may not be generalizable to some populations, as there was underrepresentation of some minority groups and older patients in the trial. Next slide, please. After reviewing the data, we gave lecanemab an evidence rating of promising but inconclusive. The PI rating reflects the uncertainty in the clinical significance of the findings. It allows for the possibility of a small benefit all the way up to substantial net health benefit if longer treatment with lecanemab demonstrates greater efficacy, for example. But this also includes the possibility of net harm if either if the ARIA risk is greater than in the clinical trial or the benefit is not clinically significant, so that you have net harm from the risk of ARIA. So I'll stop there. I appreciate your attention, and I think we'll move to questions.
Thank you, Dr. Lin, for this outstanding review of the clinical evidence and this presentation. We have about 20 minutes budgeted for questions from any CTAF member, patient, or clinical expert. Please use the raised hand function and I will call on you. I'm going to start with a question for you, Dr. Lin. Can you say a little bit more about age? I saw the mean age for the participants was 71, and you mentioned that lecanemab seems to be less effective in patients who are younger. What was the age range in the trial? Was there an upper limit, and any other factors about age in the outcomes?
Yes, thank you, Dr. Fox. So the inclusion criteria for the clinical trial had between 50 and 85 as the upper limit for age. And if you look at subgroup data, and actually, if we could go back to the slides, we have a slide actually of the subgroup data. Let's see if we can go those slides. Anyway, while we're pulling up the slides, in the subgroup data, there we go. Yasmin, I think it's slide, let me see. Slide 35, I think. You can find that. No, go one more slide, sorry. That one, yes. So you can see here that this is subgroup data, and people, these last three lines, people less than 65, 65 to 74 years, and greater than 75 years. And here, to the left of this center line, favors lecanemab, and to the right favors placebo. And you can see here that actually the younger patients have a non-significant change in CDR-SB compared to those over the age of 65, and especially those over the age of 75. And I don't know if our clinical experts want to give any context, additional context.
I think these forest plots are all notable, depending on the data surrounding the forest from other studies. The ability to make conclusions from them is strengthened or weakened. This age finding is notable for under 65. I wonder, and the table can't let us know this, I don't know if the data are available, how much that may reflect that people who have an age of onset under 65 are often APOE4 carriers, particularly homozygotes, and it may reflect the findings that we see from the homozygotes as well. So I wonder to the degree to which there may be consanguinity, if you will, between this age finding and APOE4 carrier status.
Thank you. Unless there's another comment from you, Dr. Lin, I'm going to move on to others who have questions. Okay. Dr. Murphy.
Yeah, thank you. I have two question comments. So, your view of the effect on disparities is optimistic, assuming there's increased screening for early stage disease, but the converse of that is if that doesn't happen and we were to decide that this drug is effective, then there would be greater disparities than already exist. The other question I have, or comment, well, this is a question, is related to the progression. How many people during the course of this 18 months progress to outside of these mild stages of Alzheimer's disease, and in the follow-on trial as well? And were the people, for example, that had the fatal bleeds at a later stage of Alzheimer's disease. What I'm trying to get at is, it seems that the risk from historical data is greater in using these, using anti-amyloid agents in later stage disease. So if we're looking at this as a long-term therapy, is the risk of harm going to be greater as patients progress through the disease?
Thank you, Dr. Murphy. So we don't have data to know how many people progressed. We do have, and Dr. Whittington in the modeling will talk about this, we have some data that the hazard ratio for progression to the next stage of disease was 0.69. So about a 30% less probability of progressing to the next stage of disease, whether MCI to mild or mild to moderate Alzheimer's during the 18 months of treatment. But we don't have other data to understand who exactly transitioned and who did not. And we also don't have a lot of details about the ARIA and it broken down and the bleeding by age and stage of disease. So unfortunately, we just don't have those data.
Okay, and they don't, and just to be clear, they don't say what percentage of people progressed, just that there was a difference in progression.
That's correct.
But clearly those data exist.
If I may, there's an appendix that accompanies the Clarity AD publication, the Van Dyke et al. And in the supplement, I would look at figure S6, which is time to worsening on the global CDR. And after 18 months of treatment, you have to draw, it looks like about 32% of individuals progress from either 0.5 to 1 or 1 to 2 so on the global CDR. And in contrast, on drug, about 24% after 18 months progress from 0.5 to 1 or 1 to 2 on the global CDR. It's figure S6.
And those are the cut points for the different stages?
The CDR global is a staging that, it's actually how a CDR was originally kind of conceived. The sum of boxes is a curious reuse of the CDR. So the CDR is 0.5, 1, 2, or 3. There's another version out which has an additional profound and terminal stage. Anyway, in this study, the majority of people were CDR 0.5, about 80%, and that would be progressing to 1, which is going from very mild to mild, and then 1 is going from mild to moderate stage dementia.
I just wanted to comment also, though, on the concern about development of ARIA later on. Most of the ARIA you see is pretty early with these therapies. So while I don't know that we can be sure that as patients progress to moderate or severe, if you continued the drug, that new ARIA couldn't develop, the ARIA tends to happen in the first 6 months with the lecanemab.
I'll keep us moving with other questions. Dr. Redberg.
Thanks, Rena. Excellent presentation, Grace. I have one clarifying question and then 2 specific. So to qualify for this trial or to be diagnosed with Alzheimer's disease, do you have to, in addition to mild cognitive impairment, have to have shown abnormal protein deposits, or is the diagnosis made just on the basis of mild cognitive impairment, which seems very common as people age, they forget things, names, keys?
Yes, thank you, Rita. Yeah, you did have to have amyloid, evidence of amyloid. So that's either via a PET scan or evidence of amyloid in your CSF.
Thanks. And the other, usually we're looking at 2 trials for a new drug. Was there just Clarity, and is there plans for another RCT?
So you're correct. It's only Clarity AD, and as I understand it, there isn't another phase 3 that is going to be done prior. The company has already filed for full approval of lecanemab using the Clarity AD data as the basis for its application.
I would say that's disappointing because there is a reason for 2 to assure us of findings. And my last question is, there are, as you know, other Alzheimer's treatments, as you mentioned. Can you compare the change in cognitive function, say, with Aricept, and also compare lecanemab to aducanumab? So two questions there.
Yeah, so that's a great question. So actually the change in CDR-SB in the lecanemab trial looks very similar to the positive aducanumab trial. You have to remember there were two trials in aducanumab. One was positive, one was negative. So this looks very similar to the positive aducanumab trial. And overall, anti-amyloid trials, again, as I mentioned, there's a lot of inconsistency, whether you see changes in CDR-SB or not. In terms of comparison to the older Alzheimer's drugs, I don't have the numbers off the top of my head, but these changes are kind of in the same ballpark as the changes that you would see with donepezil, for example, some of those trials that randomized trials that looked at 24 weeks of treatment with donepezil. So they are, kind of in the same ballpark.
That's
So,
very helpful. Thank you.
Could I comment there?
Sure, go ahead.
Yeah, I agree that the magnitude of the effect is similar. I think perhaps donepezil and memantine were a little bit larger, but they were conducted in different patient populations in a different era and a background of different therapies or absence of therapies. And so it's difficult to compare directly. I think it is true that currently approved medications have a fairly modest benefit, but a direct comparisons without a head-to-head trial really, it's difficult to do that.
Yeah, just to add,
Certainly some of these patients were on donepezil, it seemed like 15%, and I don't know how they did compared to the others in the trial.
If I might make one other point, Jay, and that is there were a lot of people on these other approved medications during the trial. Some of the people on these medications didn't have a clinical diagnosis of dementia with Alzheimer's disease. They have mild cognitive impairment. And to me, that's an indication that a lot of medicines are given off label. These drugs aren't approved for mild cognitive impairment. And, one can't expect the labeling to be followed for this drug or any other drug going forward. Once it's approved, then physicians are free to prescribe as they feel best for their patients.
yeah, many of the patients were on those quote background therapies of donepezil and what other cholinesterase inhibitors. So these study results reflect this drug on top of that. The second point is that the Alzheimer's disease cooperative study of donepezil and MCI failed to show donepezil beneficial in people with mild cognitive impairment. It was a negative study, goes back 15 years ago in the New England Journal, days of old. And so, and while people may off-label use, as Victor has pointed out, Dr. Henderson pointed out, it's not labeled for MCI. In addition to the MCI that's been studied in persons without confirmation that they have elevated amyloid, as is the case in this study. And again, the majority of the people in this study had MCI, CDR.5, and 65% of them had MCI.
I'm going to move on to Dr. Melnikow's question, and looking at the time, I want to make sure that we get to everyone. So we'll try to keep each one to 1 or 2 minutes.
Okay, thank you. Thank you, Dr. Lin, for a really excellent presentation. And I have, a quick comment now and a question. So my comment is it would be very interesting to see, in the light of the last conversation about the other treatments, some kind of a stratified analysis about the effectiveness of lecanemab based, on whether people were receiving these other treatments or not. But my question is about the actual difference in the main outcome variable, and whether you could give some kind of real world example of what the level of difference that was found in the trial would mean in terms of this scoring system.
That's a good question. I actually will invite our clinical experts who see a lot of patients with dementia to kind of put that into context.
So it depends a little bit on where in the CDR you are, in the sense that the CDR is made up of subscales with descriptive labels. And looking at the labels, one might find going from one box to another a difference that might be clinically meaningful. But overall, a 0.5 point in an 18-point scale, looking at the scores globally, I think really is not, the differences really aren't discernible. I think you can pick and choose within the CDR and find places where you think the differences might be meaningful, but I don't think you can extrapolate that to the overall findings.
Yeah, one overall point, the CDR sum of boxes is rarely, if it ever, used in expert practice. It's a research instrument. Having stated that, the CDR as a global concept is frequently used. That is to say, the idea that a person is very mild, moderate, severe. While the CDR sum of box scale is an 18-point scale, because, as I say, 80% of the people in this study had a CDR of 0.5, the majority had, therefore, a sum of boxes score that was very much bundled around scores of sum of boxes scores of 0.5, 1, 2, such that the mean sum of boxes score was 3.2. So you've got a very bundled group far on one end to that 18-point scale. Hence, this difference actually starts to become interesting because everyone's sort of bundled around that as opposed to spread out across 18 points. In the movement from being 0.5 to 1, a change in of 0.5 on the CDR can be significant, moving you from, and these are judgments by clinicians, I'm laughing a bit, benign forgetfulness to clinically significant forgetfulness. That'll give you a 0.5 and bump you over on the CDR.
Okay, thank you. Dr. Druva.
Thanks so much, Dr. Lin, for your excellent presentation. My question is related to blinding and thinking about the rigor of the Clarity trial. So you mentioned that ARIA was more common in patients in the lecanemab versus the placebo arm, which could result in, for example, functional unblinding. And I'm wondering if there was any impact, if there was any potential impact, is there any way that we can sort of gauge if that was related, if there was any impact of the functional unblinding? And similarly, were there other, for example, clues like infusion reaction differences among lecanemab versus placebo that again could have led to any functional unblinding? Because when I look at the endpoints in Clarity AD, I think that obviously blinding is incredibly important to being able to understand how rigorous they are. Thank you.
Yeah, thanks, Dr. Druva. That's a great question. So you're right that there are a lot of potential ways for functional unblinding to occur. There were some infusion reactions. Infusion reactions were common, and I don't have the exact numbers, but they were common. And then, as you say, with ARIA, you need additional MRI imaging to monitor, and you may actually even stop dosing. So it would be ideal if you were really to want to blind people to which group they were in, that you would have sham ARIA, for example, or you would have some people in the placebo group also undergo more imaging, more MRI imaging, for example, to mask that effect. The experts that we talked to didn't seem to think that functional unblinding was a huge issue in this trial. But again, these are small differences on the CDR-SB scale, so it may be a little bit hard to discern how much impact that had.
So if I might comment, I think there would have been some unblinding. That's unavoidable. I think if the effect size were larger, then the effects of the unblinding really wouldn't matter very much. But when the overall treatment effect size is small, it's difficult to exclude placebo effect as contributing to that small difference. So again, with a larger effect size, I don't think the degree of unblinding would have had an important impact. Here we can't judge. It may or may not have.
Thank you so much.
The Van Dyke study, just to add on this one, but the Van Dyke study, again, the Clarity study, the phase 3 study, phase 3 by regulatory filing, reports sensitivity analyses to address issues of dropout as well, and also the relationship between incidence of side effects, and particularly ARIA, which would be what could lead you to be unblinded, and impact on outcome. And the report is the sensitivity analysis did not show a change in the outcome analyses.
Okay, next question, Dr. Phillips.
Yeah, thank you very much. It would seem really important to have patient level data So I didn't understand why we don't have it, and will we have it?
Thanks, Dr. Phillips. Yes, we don't have it at the moment. And whether or not we will have that is a question of whether the manufacturer will release the data from the trial. But as of yet, they haven't published anything on an individual level. And I don't believe that in the published data, there are any published individual level analyses from anti-amyloid drugs.
If you mean that generically, I think during the FDA presentation for aducanumab, which doesn't necessarily apply here, that they didn't find a correlation between the amount of amyloid removed and the CDR sum of boxes, but that was a different drug, and that was in the FDA report and not in the published data, as far as I'm aware.
That's correct. That's what I picked up from the statistician's report. I kind of set aside discussions of aducanumab given the complexities of the study's design, interpretation, et cetera. I really just view that as not apples and oranges, but I don't know, tennis ball and apple comparison. They're round, but otherwise they're totally different. I think the more compelling biomarker data with this drug are the data around total tau, phosphorylated tau, and progranulin, which are measures of neurodegeneration.
We are going to allow a few extra minutes, so I'm going to call on Dr. Lee, and then we'll probably have time for maybe one or even two more if they're short. Okay, Dr. Lee.
Thank you, Dr. Fox, for allowing me to sneak in under the wire. One of the things that I was thinking about that's challenging about this disease and inclusion criteria is that not everybody with amyloid ends up developing dementia. Not everybody with amyloid and even some cognitive deficits end up progressing. And even though the placebo group shows an average decline, I was wondering if either Grace or our clinical experts could talk about the proportion of patients who would meet the inclusion criteria of this study who would actually not be expected to progress. And this would be a population potentially that we would be exposing to the risks with little chance, with almost no chance that they would benefit. So I don't know if this is I guess bringing in, given that we don't have individual patient level data on the studies, I don't know if we can use the study data to answer this question, but maybe natural history data. But that would be really helpful for me.
Yeah, that's a great point, Dr. Lee. Yeah, there is no study data that would help answer that question because we would need the individual level data. I think the other interesting thing here is that 7%, almost 8% of the placebo group cleared their amyloid as well. And so it's sort of unclear. And I think this goes back to the uncertainty of what role does amyloid play and how closely is it correlated with cognitive outcomes. I don't know if there's any clinical data, any data in the clinical trials that we have that could answer that question. And Dr. Henderson or Dr. Karlowicz, do you have any other comments?
I think that there's some natural history data through ADNI, the Alzheimer's Disease and Neuroimaging Initiative, which of course is a voluntary cohort as well. And I actually don't know the results exactly. I think, as you're saying, it's very common for people without cognitive impairment after age 70 or so to have amyloid demonstrable by PET imaging, amyloid PET imaging. I think that those people are at increased risk for progressing to dementia, but I don't think the follow-up has been long enough to know whether all of them eventually will, whether a substantial proportion won't. I don't think that information is known, but that would be one place to look for at least a partial answer.
Yeah, I would entirely set aside the discussion of non-cognitively impaired individuals with amyloid. These individuals had cognitive impairment, and ADNI data and other data for individuals with cognitive impairment by metric criteria on measures like logical memory therefore meet criteria for MCI or mild stage dementia with elevated amyloid are at substantially increased risk of progressing. I think baked into the question about a non-progressor is this someone who has just very slow disease, or almost, I'm wondering, see if you're thinking just doesn't have the disease. And I think one of the things that I think the field is struggling, well, struggling is not the right word, is wants to learn more about is with these data, is how folks with amyloid and tau responded to this drug. Tau meaning phosphorylated or total tau, what the relationship between those two biomarkers that pathologically define the disease and response to this therapy are. And under the ways these studies are done right now, that wasn't sort of the way the data were analyzed. The data are there though to look at. All the tau data are there, blood and spinal fluid.
I do want to allow Dr. Langer Gould's question. Can you be very fast?
Yes. Victor and Jason, and I guess Grace also, reversal of MCI. I know there are natural history studies, all cause MCI of a 10-15%, I believe, that will go back to having normal memory. How does that apply to people with Alzheimer's disease as their etiology of MCI? Is there any data? And was there any findings in this study of people either in the placebo group or lecanemab actually improving and going from MCI back to normal cognition? Or are we only talking about every single person in this trial worsening, but at slightly slower rates?
So I think depending on the cohort that's being followed, there are a lot of people that transition in both directions. And that's not surprising. There's diagnostic error, there's measurement error in the psychometric measures. Yeah. I think one of the cohorts is from the National Alzheimer Coordinating Center data set representing all the Alzheimer's disease research centers, where a substantial number of people classified as having mild cognitive impairment ended up not becoming demented, ended up transitioning back to normal. But I would attribute that primarily to a measurement error. And I think that would be present in any sort of study, including a clinical trial like this, that given the measurement uncertainty, there would be people who would be sort of close to a boundary and would transition over during the next measurement period. And that's also true for imaging measures as well, I think.
Yeah, I think if you define MCI by metric criteria only in a population-based study, you will see noise and state transition. If you define MCI based on a clinical interview by a clinician, such as a CDR interview, I can't say what the sort of still the transition rate back because of issues of error and whatnot are, but it'd be substantially lower than if you simply just administered a series of metric scales with cutoffs and said you've got MCI. So sort of the difference between cohort study classifications versus clinical classifications. I think the addition in this population of the requirement for elevated amyloid for entry into the study and randomization adds to the likelihood that those who had MCI were individuals where there was cognitive impairment as opposed to measurement error. But issues of measurement error always, of course, always still aren't there, if you will.
All right. Thanks, Dr. Karlowicz and Dr. Henderson. I think, unfortunately, we need to move to the economic modeling. This has been a great discussion. So I will now turn over the platform to my colleague, Dr. Melanie Whittington, to go through the presentation for the economic model.
Thank you, Dr. Lynn. Hi, everyone. I'm Mel Whittington, Director of Health Economics at ICER, and I'll be walking through our economic model findings. Next. I have no conflicts to disclose. Next. The objective of this component of our review was to estimate the lifetime cost effectiveness of lecanemab in addition to supportive care as compared to supportive care alone for the treatment of early Alzheimer's disease. And supportive care can include non-pharmacologic and pharmacologic, but not disease-modifying therapies. To achieve this objective, we created a Markov model from the United States setting. We present two perspectives side by side, our healthcare sector perspective that emphasizes direct medical costs and consequences to the patient, and then a modified societal perspective where we include additional elements, including patient productivity, caregiver quality of life, caregiver direct medical care costs, and caregiver time spent caregiving. Our time horizon is that of a lifetime with a discount rate of 3% per year for all future costs and consequences. Our cycle length was 1 year, and our primary outcomes are cost, quality adjusted life years, equal value life years, and years in the community setting. In this presentation, I'll be emphasizing cost, qualities, and equal value life years, but I do encourage you to look at the report for all of the outcomes. Next slide. This slide shows our model schematic. Each of the ovals represents a health state of our model, and so you'll see it's defined by Alzheimer's disease severity, where we have an oval for MCI due to Alzheimer's disease, mild Alzheimer's disease, moderate Alzheimer's disease, and severe Alzheimer's disease. The cohort can transition to the dead health state due to all-cause or disease-specific mortality. And the arrows represent all of the possible transitions between these health states. Now, it's not marked here as a health state, but certainly tracked within the model is the setting of care. So we tracked whether the individual was in the community setting of care or the long-term care setting. And once individuals reached the long-term care setting, they stayed there until death. Next slide. The starting population for our model included adults with early Alzheimer's disease, defined as MCI due to Alzheimer's disease or mild Alzheimer's disease, with confirmed amyloid positivity prior to the start of the model. Our mean age and percent female are in the table on this slide, as well as the baseline clinical stage and setting of care. So at baseline, 55% were in MCI and 45% were in mild. That estimate is not from the trial, but from a population level registry. Importantly, the baseline starting age were those two clinical stages, but over the lifetime course of the model, the cohort could progress to more severe levels of Alzheimer's disease. The baseline setting of care, 92% in the community, 8% in long-term care. Again, over the lifetime time horizon of the model, could transition to long-term care, to which they would stay there until death. Next slide. I want to emphasize a few of our key model assumptions, the first of which we assumed that lecanemab was effective at slowing the progression of disease while a patient had MCI due to Alzheimer's disease or while they had mild Alzheimer's disease. The evidence on lecanemab is in patients with early Alzheimer's disease, and based on the trials of anti-amyloid therapies suggesting no benefit in moderate Alzheimer's disease, we heard from clinical experts it is unlikely for lecanemab to have an effect in moderate or severe Alzheimer's disease. And so that brings us to our second bullet, in that we assumed patients would stop receiving lecanemab treatment once they reached moderate Alzheimer's disease. Now, we do know there is uncertainty here, and we know this is an area of needed future research. And so we have altered these assumptions in scenario analyses that we'll present here, and it has much more detail on in a report. The third bullet, there's no clinical benefit assumed after a patient stopped treatment. Robust evidence is lacking on lecanemab's effect on clinical outcomes after a patient stopped treatment. And so for our model, we assumed no clinical benefit after treatment stop. And the last bullet, something we've already talked about a little bit today, all occurrences of ARIA and its associated consequences were modeled in the first year of treatment. And that's due to consistent findings that ARIA typically occurs early on within the first 6 months of starting treatment. Next slide. I want to emphasize a few of our key model inputs, but again, much greater detail on the sources and an exhaustive list of inputs are provided in our report. This slide reports our effectiveness on disease progression. So the most relevant clinical evidence for the model would be the rates of transitions among our health states, which are defined by Alzheimer's disease severity. And as Dr. Lin already mentioned, for lecanemab, there is phase 3 evidence on the progression to the next stage of dementia for patients in MCI and mild. And that's where we're getting the treatment effectiveness for MCI and mild. So that 0.69 is a hazard ratio on the progression to this next stage of dementia. So that evidence served as the best available evidence for us to estimate the treatment effect of lecanemab on slowing the progression in MCI due to AD or mild AD health state. Importantly, that estimate, we do not have a standard error or a confidence interval around. And so for our sensitivity analyses that we conduct, and I'll show in a little bit, we assumed the same standard error from the primary endpoint of the pivotal trial from Clarity AD. Related to the assumption I mentioned on the previous slide, we did model treatment stop at moderate Alzheimer's disease and no benefit after treatment stop. So the benefit stopped in moderate Alzheimer's disease and severe Alzheimer's disease. And that 0.69 in health states of MCI and mild were applied to progressions to more severe disease. Next slide. The model also tracked adverse events, the probability of any ARIA, the probability of symptomatic ARIA, and the probability of adverse event-related discontinuation was tracked in the model. The probability of any ARIA influenced the model by way of additional monitoring costs, so 3 MRIs for each ARIA event. The probability of symptomatic ARIA influenced the model by way of an additional disutility for 12 weeks, the average duration of ARIA. And you'll notice these ARIA numbers are a little different from what Grace or Dr. Lynn reported, and that's it's the same source of evidence. This has just any concurrent ARIA-E or ARIA-H removed. So for the purpose of the model and assigning costs and consequences, we didn't want to double count. And so given ARIA-E and ARIA-H can occur concurrently, we removed any concurrent instance to avoid any double counting. Next slide. This slide reports our health state disutilities, both for the patient and the caregiver. The caregiver disutilities were brought in the modified societal perspective. This evidence source allowed us to have one evidence source for both the patient and the caregiver, with evidence stratifications by community setting and long-term care setting. And so you can see from this slide, as severity of disease increases, the impact of quality of life becomes quite large. Next slide. A few cost inputs to emphasize. The annual wholesale acquisition cost is what we used for the treatment cost of lecanemab in our model. That is $26,500. That cost does not include any provider administered markup that might be associated with this treatment. In our model, we assumed a markup of 6%. The treatment is also associated with monitoring and administration costs. We assumed 4 MRIs in the first year of treatment, and that was in addition to any additional MRIs that might be required to monitor for ARIA. After individuals already had symptomatic ARIA, they would have additional MRIs. There's also an IV administration cost that was assumed per administration, and we did not include any diagnostic costs because we assumed amyloid positivity was determined prior to model start. Next slide. All right, I will now go over the results. Here are our lifetime model outcomes from the healthcare sector perspective. You can see in the lecanemab treated arm, it was more costly, but there are also greater health outcomes across all outcomes observed: life years, quality adjusted life years, equal value life years, and years in the community. Next. I'll orient you now to the bottom half of this slide that now displays the modified societal perspective. So again, in the lecanemab treated arm, we see additional costs, but additional life years, quality adjusted life years, equal value life years, and years in the community. When comparing across the two perspectives, the incremental costs, so the difference in costs between lecanemab and supportive care, between the two perspectives, the incremental costs are lower in the modified societal perspective, although the magnitude of each arm is larger when we're now considering caregiver time spent caregiving and caregiver direct medical costs. Also, comparing between the two perspectives, when you're looking at the QALYs and the EVLYs, the magnitude for each arm is lower in the societal perspective because now we're including that caregiver disutility, but the incremental difference is slightly larger in the societal perspective. Next slide. So I think that's easier to see when we're looking at this incrementally, and that's displayed on this slide. From the healthcare sector perspective, the cost per quality adjusted life year gained was $254,000. Cost per equal value life year gained was $204,000. When looking at the modified societal perspective, the cost per quality adjusted life year gained was $236,000, as compared to the incremental cost per equal value life year of $183,000. So all of these incremental cost effectiveness ratios are above commonly cited cost effectiveness thresholds. Next slide. We conducted numerous sensitivity analyses to assess robustness of the findings and assess variation. The one-way sensitivity analysis is a terrific analysis to identify what's really driving the model. And overwhelmingly, that was the hazard ratio on slowing the progression of disease. And so that was the main driver of this model, not surprisingly. The probabilistic sensitivity analysis is helpful to inform us what's the probability of being cost effective, accounting for all of that uncertainty in the main drivers, but also in other key inputs. The results from a probabilistic sensitivity analysis suggest that at a threshold of 100,000 for equal value life year gained, there's a 0% to a 1% of being cost effective, depending on which perspective you look at. At the higher threshold of 150,000 per equal value life year gained, there's an 11% to 25% probability of being cost effective. The results with the quality outcome are slightly less favorable than this, but one point to consider here is that standard error for our main driver, the hazard ratio and slowing progression of disease, was based on the primary outcome. We didn't have that directly for that input. Next slide. I'll present one scenario analysis here because I mentioned this while we were talking about our key model assumptions, but again, there's many more scenario analyses in our report. This one here is the treatment stop at severe Alzheimer's disease. So you can see the estimates are relatively similar to our base case. In this scenario, the incremental clinical outcomes are greater because we assumed the same benefit, the same 0.69 in moderate Alzheimer's disease as well. So we made that assumption. And so here, yes, the clinical outcomes are greater, but also the costs are also greater. Now you have larger treatment costs and larger non-intervention costs. So relatively comparable. Next slide. A few limitations to emphasize. Again, I know I've said this a few times during my talk, but the first is that the confidence interval for that hazard ratio on the progression to the next stage of dementia was not available. And also we heard from many individuals that the utility evidence is from a study published more than 20 years ago. And that is true, and I can speak a little bit to why we still decided to use that study. There is a recent meta-analysis that many individuals encouraged us to use, and we appreciate that feedback and that recommendation. We were aware of that source. The reason we didn't choose that source is only for patient quality of life. There isn't caregiver quality of life in that source, and it's not stratified by long-term care setting or caregiver setting. Additionally, that source does provide a point estimate for each of the patient health states. So it provided a utility for the patient for MCI, mild, moderate, and severe. And the range of utilities that we assumed and included in our sensitivity analyses included the point estimates from that meta-analysis. So we continued on with the utility evidence that we chose, albeit it is 20 years old. It was comparable to recent meta-analysis efforts, and it had evidence for the patient and the caregiver stratified by setting of care. Next slide. We're so grateful for the number of comments we received, a few that I will highlight here, one of which was to use a patient level simulation model like AD-ACE. AD-ACE is a patient level simulation model that some individuals use to model Alzheimer's disease. It was used by lecanemab's manufacturer in their modeling efforts. In AD-ACE, surrogate endpoints, biomarkers are used to then map to clinical outcomes and health states, et cetera. So instead of taking the indirect route to get to the health states, we just directly used the phase 3 evidence of progression to next stage of dementia in our Markov model. Second bullet here is use a threshold above common thresholds. We're certainly aware of this literature that is suggesting higher thresholds for more severe illnesses with high unmet need. I was actually at a conference last Friday with the developers of this method, and importantly, this literature also suggests lower thresholds for less severe illnesses with lower unmet need. We'll continue to stay on top of that methodological development, but in the meantime, we're continuing to present a range of thresholds from 50,000 to 200,000 per outcome gained. And the last comment that I'll emphasize here is patients with Alzheimer's disease have more than one caregiver. We certainly understand that. In our model, we did assume the costs and consequences for one primary caregiver. So I think that's what's driving a lot of those comments. A few things to note is the evidence that we use for the caregiver on our model. So caregiver quality of life, caregiver direct medical care costs, and caregiver time spent caregiving was all retrieved in those data collection efforts from one primary caregiver. It could be very different if it was a primary caregiver and a secondary caregiver et cetera. And so the evidence was for a primary caregiver that informed our model. Another thing is to remember how the caregiving impact is factoring into the model. And with lecanemab in our base case scenario, we were seeing additional time spent in MCI and mild Alzheimer's disease, which is associated still with a caregiver impact. So although it's not taking patients to a health state that completely removes the caregiver impact. And then there's also life extension here. And so the caregiver impact is complicated here. There's really two levers. Yes, there is a keeping individuals in less severe health states for longer, but that also requires additional caregiver time spent. Next slide. With that, I'll conclude that at the current wholesale acquisition cost, lecanemab exceeds commonly cited cost effectiveness thresholds. And the findings here are primarily driven by the effectiveness of lecanemab at slowing the progression of disease. And with that, I think we can take comments.
Thank you, Dr. Whittington, for this outstanding work and presentation. And we will allow around 20 minutes again for questions from CTAF members, patients, or clinical experts. I'm going to ask one to start, which is about the decision not to include diagnostic inputs. And as a primary care doctor, I don't see most patients who are diagnosed with Alzheimer's or being worked up for Alzheimer's getting LPs for CSF or PET scans. And I guess this would be a question for the experts as well, and maybe for insurers, whether we anticipate this being a requirement for use of lecanemab. But I think between referrals to specialists, LPs, and or PET scans, that would really change that cost structure.
Yeah, I might respond first, and then we can open up to the clinical experts. And I'm anticipating this might come up at the policy roundtable, so Steve can correct me if I'm wrong. Yeah, so for the model, we did not include diagnostic costs. This was because we assumed that happened prior to the start of treatment. And so our model is starting at the treatment start and assuming our patient population included confirmed amyloid positivity. So I just wanted to be transparent about what we did in the model, and then I can open it up to the others to talk about the other great contextual issues you brought up.
I guess in clinical practice, it would be necessary to obtain evidence that amyloid was present either through spinal fluid or through PET imaging. And I think for a drug that were truly effective, then this would happen. I think it would be important to do, but it's still an essential step regardless.
But in practice right now, that's not, at least where I practice, that is not a routine test that would be done for a diagnosis.
Right, and it needn't be routine except in the setting of consideration for a treatment like this. Then it becomes routine for that specific purpose.
Thank you. Okay, let's call on Paul.
Yeah, thank you for that. And I understand the modeling and why, if we believe in this hypothesis that this would all be progressive, but I think the assumption of the mortality benefit is a very strong assumption. And I know you probably haven't done these exact calculations, but if we just assumed the observed quality of life benefit, put that into utility, and then assume that lasted for some limited a period of time afterwards, I would imagine the cost effectiveness numbers would be, several orders of magnitude higher, right? It would be it wouldn't even be approaching what we might consider an upper level of cost effectiveness. What are your thoughts on that?
A very interesting question. If you look at the model outcomes, you're right, there is this life extension that was associated with keeping individuals in less severe health states. And there was how mortality was modeled in the model is for each health state, there was a standardized mortality ratio. So that number was higher for more severe health states. And so indirectly, by keeping individuals in less severe health states, we see this survival gain. So it's hard to disentangle mortality from health state occupancy, but I can say out of our value-based price or our the value attributed to this to this treatment, life extension and quality of life factored into that about equally.
Dr. Redberg.
Thanks, Rena. And maybe cardiologists think alike, because my question was also, I was very concerned and struck by that benefit on life years when we didn't see any mortality benefit. And in fact, we saw 3 deaths in the open label extension in the lecanemab arm. Did you account for the deaths in any way? I didn't see that in your model. And then I have a second question on utilities.
Great question. We did not capture any death. We only we modeled death as all cause and disease specific death, not the direct deaths that were reported.
Right. So you modeled a benefit, but actually what we saw were 3 deaths in treatment arms. So I am concerned about the reliability of that model. And the other thing is, did you model disutilities for having a biweekly injection and having to get a lumbar puncture and having to get PET scans and other things?
Great question. No additional disutilities were added for that. Clinical experts, does anybody want to comment on the first question she asked?
In an Alzheimer's study measuring a benefit of an intervention, I would be almost existentially astounded if mortality was measured as a benefit, meaning reducing mortality from the disease, quite a contrast to cancer and heart disease.
That makes sense.
Yeah, it would. I mean, I said existentially to hammer home the point. I mean, living longer with this disease is incredibly morally problematic, depending on the stage of disease. And I make no judgment on that. I simply offer that as a very plain standard observation. It's so different from other chronic diseases of aging.
Right. And just the point was made, but the difference in the 2 groups wasn't that one got better. It was that one deteriorated at a 2% lower rate than the other one.
Right. 2% on an 18-point scale with everyone bundled up on one end, though, Rita.
Yeah.
And slowing of a disease like many cancers and neuroinflammatory diseases is certainly a well-described paradigm. And cardiovascular disease, people still die of heart attacks, just not as they don't die as soon after their treatments for their various cardiovascular causes of heart failure and et cetera.
And I'll just comment, I was concerned that there was no benefit in the women in the trial. And I don't know if you modeled the cost effectiveness separately by men and women.
We did not do any subpopulation.
Thank you.
Thank you.
Dr. Murphy.
Yeah, given you said that the hazard ratio was such an important driver of this and there were no error bars associated with it, did you play around to see what it would have looked like with a hazard ratio, say, of 0.79 or 0.59 as opposed to 0.69?
Great question. In our tornado diagram and using the standard error from the primary endpoint of the trial, we varied that from around 0.5 to 0.9. So that range in was captured in our one way and in our probabilistic sensitivity analysis. It was essentially 0.5 to 0.9.
Okay.
Dr. Lee.
Dr. Whittington, thank you for these results. These are really interesting. One thing that I was kind of struck by, and I think this is similar to what Dr. Heidenreich and Dr. Redberg was saying, is that I'm struck by the fact that what we have is data on the transition from like MCI or mild to moderate, but so much of the results are being driven by the transitions from moderate to severe and severe to death. And so it makes me a little bit, and I think some of the assumptions that you mentioned, I think for the, I guess the boosters, one could imagine, hey, if we are true believers of the amyloid hypothesis, maybe getting rid of amyloid, which everyone agrees this drug does, actually would have long-term beneficial effects. The other counter, the non-anti-boosters, I guess I would say, could argue, you know what? We call it area E and area H, but when you talk about brain bleeding and brain swelling, I worry that there may be long-term adverse effects about that. And it seems like brain volume is getting lower. And so you could imagine long-term adverse effects with this treatment. Both of those are just completely unknown at this point. And I worry a little bit about how much uncertainty there is going to be in the results, because so much of those results are driven by those late terms. So I guess my question after that long monologue is there a way that we can decrease the window of the analyses to say within a year or 2 or 3, within the observation period of the study, what are the most likely cost effectiveness results? I don't know if this is something that is ever done, but I just want to try to get a sense of so much of the results, the lifetime results, feels like based on assumptions which we really don't have good data on.
Sure, there was a lot there. A few comments. Let me know if I don't answer it adequately. The long-term results, once an individual in either arm has reached moderate Alzheimer's disease, they're going to follow a similar course between the lecanemab arm and the comparator arm. So really the action is what's happening in MCI and mild, those populations that we did have in the clinical evidence. So yes, we are extrapolating over a lifetime time horizon. That makes assumptions, that we're required to make assumptions there, but our assumptions are linked to health states, and so and time in those health states. I'm not trying to undersell that there's not a lot of long-term assumptions there, but a lot of the action that's differentiating the lecanemab arm and the comparator arm is occurring in that MCI and mild health state.
Your model had therapy ceasing at a transition to CDR2, isn't that correct?
Yes, at moderate Alzheimer's disease.
Right, the presumption would be ceased.
Mm-hmm. So with treatment stopping, clinical benefit stopping, the difference between, in the late stages of Alzheimer's disease, we are not modeling any difference between the arms. But yes, enormous uncertainty that only so much can be modeled in.
Yes. Tony.
Yeah, thanks, Dr. Whittington. Could you clarify if the caregiving costs that were include some value of family members? And assuming they do, how did you value that?
Yes, could you expand what value of family members? I can tell you that we included primarily time, uncompensated time spent caregiving, and then a quality of life decrement.
It was included.
Mm-hmm.
Okay, thank you.
In the modified societal perspective only, for the primary caregiver.
And how did you come up with a value for that?
Luckily, I didn't have to. Luckily, there is literature, but there is. This is a very important area for further research and methodological development, capturing utilities for caregivers and for patients with Alzheimer's. So the source that we used used the Health Utilities Index to capture those utility estimates for the caregiver. There is in our report a scenario analysis that used a very different approach. It uses a time trade-off exercise among individuals of the general population, asking them, hey, put yourself in the shoes of a caregiver of Alzheimer's disease. What would the disutility look like? The estimates are quite large and different from what we are currently doing. That did not become our base case analysis because that's reviewed. It's not peer reviewed. There's a lot of questions about the appropriateness of that method. The questions didn't map exactly to the health state. So that's an area where there is going to be a lot of work done in the near future, I believe. I'm excited for that to be done. The evidence that we're using, we believe is the best available evidence. And then we varied it across a wide range in our sensitivity analyses, and then actually updated the entire source in a scenario analyses. So larger caregiver disutilities would result in slightly more favorable cost effectiveness estimates, but they were still above commonly cited cost effectiveness thresholds.
Okay, great. I think we have time for one more question, Dr. Druva.
Thanks so much. Thanks so much, Dr. Whittington, for your expertise. My question is about the assumption that patients would cease therapy at the time of progression of moderate Alzheimer's. And perhaps a question for you and perhaps our clinical experts. I'm a little bit concerned that, and just perceiving in a patient clinician discussion that we say, well, now you've progressed to moderate, we're going to take this treatment away. I assume that's going to be a really, really hard conversation with the patient, their caregiver, and we're talking about a pretty high cost drug. I'm wondering if you might be able to sort of weigh in on that. I know it's hard to chew on, but any guidance on how we should think about that would be helpful.
Yes, that's a great question. And I think the clinical experts will probably be able to answer that better. So I might not use the time and pass it on to them.
I think in a real world setting, the way it would play out is that there would be follow-up PET imaging, for example, to see whether or not the amyloid burden had gone to zero. In that case, treatment might be stopped at the end of 18 months. If amyloid was still visible, then I think perhaps treatment would be continued based on, just what the, well, I mean, that would be my expectation. That would play out in that way based on imaging results at some point during treatment.
Just to add to that, I think that measures, particularly blood measures of amyloid and tau, but particularly tau, neurogranulin and related markers of neurodegeneration will be keenly watched and inform a conversation. I'm kind of keen to see what the extension study data show in this, for example. Having said that, you want to begin with data, but obviously this conversation will then walk into some rather ethically and morally charged matters. There's no question.
And just to be fully transparent about the model, in our scenario that I presented, we included treatment continuation through moderate, but also the exact same treatment benefit. We did not run a scenario where treatment continuation occurred and treatment benefit stopped. That would be less cost effective than that scenario because the benefit would go away and cost would persist.
Thank you. I do want to keep us running on time. I know that Russ had a hand raised earlier, so if you have a fast comment to add, otherwise we will move on. Go ahead.
Yeah, just very quickly, I put this in the chat, but this idea that it's an 18-point scale and we should think about 2 points being, or whatever, being 2%. It's not a linear scale. It's not an interval scale. So that's just not the way the scale works, and we shouldn't think of it that way. That's just, we need to be based in facts and not how we think maybe other scales work.
Thank you for all of the outstanding questions and comments raised during this discussion. And all of the questions raised are outstanding, and I know we didn't get to everyone, but please hold questions for the voting session.
Rena, sorry, let me interject just because Doreen also had a comment, and I think it would be nice to hear from her before we move to the formal public comments.
My apologies. Yeah.
Hi. As a neuro nurse, I follow the data, and for the first time in 8 years, there was hope. What I'm looking for as a person living with the disease is not to live longer. I'm going to be 70 in 2 weeks. What I want is for my tomorrow to be my today, for however long I can do that. And I am a single woman. I don't have a caregiver. I will never have a caregiver. So it's imperative to me that I can stay who I am today, tomorrow, whether I live a year, 6 months, 20 years, 100 years, God forbid. So that's, I have rheumatoid arthritis. I've been on a monoclonal antibody for that. I discussed the risk and the benefits, and we've made it what I think is a wise clinical decision. And I want that opportunity. I'm looking at the side effects, and certainly they're concerning, but that's a possibility. What I'm dealing with is the reality of what I'm living now. And I, as an individual, if I'm indeed an appropriate candidate, I'm willing to take that risk, because to me, that risk, however not tiny, is far outweighed by the higher possibility that I'm not going to be the person I am today in a week from now or 6 months from now. When I was diagnosed 8 years ago, I never thought I'd make 70, or I thought I wouldn't know that I was 70, and yet I do. And I want that possibility to be when I'm 72 and 75, and 80. After that, we'll see what it goes, and that's what we're looking at. I understand data points. I understand their check boxes. I understand all that. And while it's important when you're looking at something globally and making decisions, I think when you're looking at this particular disease, if you're looking at 6.5 million people, you're looking almost at 6.5 million variations. I am one of those variations. And to me, it's important. Now, there are 6.4 million may say no, but that's what we're looking for. It's just the chance of hope.
Thank you, Doreen. I'm really glad that we got to hear from you, and thank you for sharing those points. I will turn us to the final section of the morning for public comments and discussion. We have 5 scheduled comments today. I will introduce each speaker